Hey everyone! In this video, I'm going to talk about control of gene expression. In other words, what proteins to make and when. And it's important to remember that all cells have exact copies of your DNA, but not all cells are the same. Some are liver cells, some are teeth cells, some are eyeballs. So how do the cells become specialized? In other words, how do they make the proteins necessary for them to be the types of cells that they are? So there are several levels of gene expression or control of gene expression, but I do want to make sure that we also understand that there are some proteins that are made in all cells all the time. We call them constitutive proteins. A great example would be the enzymes for glycolysis. Since glycolysis is necessary at all points for the cell, these genes would be turned on all the time. So when we control gene expression, there are 4 points or 4 levels of control in eukaryotic cells that we're going to discuss. The first is which it happens at the transcription level. Now remember, in transcription, we are making the mRNA, and this is probably the most common type of genetic regulation. Now, at the level of transcription, we can increase the level of transcription or make it more easier for the RNA polymerase to find the promoter site by using either transcription factors or enhancers. The next level of control would be post-transcriptional. In other words, after the primary transcript has been made. So with post-transcriptional control, we were referring to a couple of things. First would be taking that primary transcript and modifying the edits that we make in RNA processing. In other words, as we take that primary transcript and convert it into the mature mRNA. So one of the changes that we make to that mature mRNA is the removal of those introns. And with this removal of the introns, we can actually change which introns and even which exons are going to be removed from that primary transcript. Another way that post-transcriptional control can be demonstrated is the 5' cap or the 3' tail. More specifically, the length of these ends of the mRNA. Basically, the longer the 5' cap and the 3' tail are, then the longer the mRNA can exist before it degrades in the cytoplasm. And the longer the mRNA is there in the cytoplasm, the more protein is going to be made. The last post-transcriptional control we're going to talk about are microRNAs or small interfering RNAs. And what these are RNA molecules whose responsibility it is to break down mRNA once it's in the cytoplasm. So the more active these are in the cytoplasm, the less protein will be made because there's less mRNA available. The next type of regulation is transitional control. So this is at the point where the mRNA is now in the cytoplasm and has found the small subunit of the ribosome, and now we're going to start making the protein itself. So a good example of this is actually physically blocking the ribosome from being able to read the mRNA. And a molecule that would do this is microRNA. So microRNAs can not only help to decrease the amount of protein made by chopping up the mRNA, but they can also decrease the amount of protein made because they will physically block the ribosome from bonding with the mRNA itself. The next topic is post-translational control. So this is after the point of translation and your amino acids have been linked together to form the protein. Now you can control whether or not that protein is functioning. So I'm going to remind you that back in Unit 2, we had learned that the Golgi body or the Golgi apparatus modifies and ships out protein. Well, the modifications that can happen is to ensure that the protein has folded correctly and is functioning. Another form of post-translational control is the process of ubiquitination. What happens with ubiquitination is that the proteins are going to get tagged with a protein called ubiquitin. And what that basically does is mark the protein for destruction. So once a protein has been tagged with that ubiquitin, those proteins are going to be destroyed and will no longer be affecting the cellular processes. The last type of post-translational control is having proteins already pre-made and packaged into vesicles. This way, when it's time for the proteins to be released or secreted or fused with the plasma membrane, they are much quicker and faster because they were already pre-made and loaded up into vesicles just waiting for the signal. The last type of control is called chromatin restructuring or remodeling. This is referring to how easily can the DNA be accessed as it's wrapped around the histones. The looser the DNA is around the histone, the easier it is for the RNA polymerase to access the genetic code and make the mRNA. The tighter the DNA is wrapped around the histone, the less mRNA is going to be produced. Now, this is a whole other section called Epigenetics, and we will go into detail about how we can wrap the DNA tighter around the histones or make the DNA looser around the histones in that section. That's all for control of gene expression. Thanks so much for watching. Bye.